Oxygen plays critical role in cancer metastasis. This is from an article printed in Genetic Engineering News, August 3rd, 2016. Scientists from Johns Hopkins University and the University of Pennsylvania reported on work that showed how sarcoma cells in mice pursue a path toward greater concentration of oxygen, almost as if they were following a widening trail of breadcrumbs. That path is suggested to lead the cells to blood vessels, through which the cells can spread to other parts of the body. If you think about therapeutic targets, you could target this process specifically, said Sharon Gurich, PhD, professor in Johns Hopkins University's Whitting School of Engineering's Department of Chemical and Biomolecular Engineering, and the lead author of the study. Intratumoral oxygen gradients mediate sarcoma cell invasion. She acknowledged that clinical application is a long way off, but said these results provide clues about a key part of the life cycle of soft tissue sarcomas and also a proven way to test cancer treatments in the lab. Cancers of all sorts are known to thrive with little oxygen, and researchers have looked at the role of low oxygen conditions in tumor development. Less well understood is how cancer cells respond to varying oxygen concentrations in their early stages. This was the focus of this research. Dr. Gerich and her team tracked thousands of early-stage cancer cells taken from mice as these cells moved through a mock-up of bodily tissue made of clear gel in a petri dish. The hydrogel replicates the environment surrounding cancer cells in human tissue. Quine Min Park, PhD, then a postdoctoral researcher in the Johns Hopkins lab, developed the hydrogel cancer cell system. And Daniel Lewis, a Johns Hopkins graduate student, analyzed cellular migration and responses to rising oxygen gradients. For this experiment, the hydrogels contained increasing concentrations of oxygen from the bottom of the hydrogel to the upper layer. That allowed researchers to track how cancer cells responded to different levels of oxygen, both the tumor and within body tissues. Analysis of sarcoma tumors in mice, for instance, shows that the largest tumors have a large area, very low oxygen at the center. Smaller tumors have varying oxygen concentrations throughout. The researchers' first step was to show that cancer cells migrate more in hypoxic hydrogels, as compared with hydrogels containing as much oxygen as the surrounding atmosphere. They then looked at the direction of the cell movement. In the hydrogel, which mimics the oxygen concentrations in smaller tumors, cells were found to move from areas of lower oxygen to higher. Researchers also found that the medication minoxidil, widely used to treat hair loss and known by its trade name Rogaine, stopped the movement of cancer cells through the hydrogel. Cancer cells are known to modify their environment to make it easier for them to move through it. But this study takes that understanding a step further, noted Dr. Gerich. We did not know that it was the oxygen that effectively directs the movement, she said, suggesting that oxygen gradient affects early stages of the metastasis process. The study also demonstrates the three-dimensional hydrogel model as an effective tool for testing cancer treatments in the laboratory, the authors wrote. Dr. Gerich said the human patient's cancer cells could be placed into the hydrogel just as the mouse cells were, allowing clinicians to see how they respond before treatments are given to the patients.